Welcome students. This video lecture is about stomata. So what are stomata? They are nothing but just a pore or opening in the epidermis. They are some space guarded by the guard cells and subsequently by subsidiary cells. So it is not any structure that is called stomata, but an opening or a pore. And in plural it is stomata and singular it is stoma. So stoma has a pore guarded by two specialized guard cells which are guarded by subsidiary cells. So this is the structure. You can see the guard cells are having chloroplast. They are guarding the pore, and this pore is your, this empty space is your stoma, and these are the guard cells having chloroplast. All the other epidermal cells don't have chloroplast, and in turn they are surrounded by subsidiary cells. So this whole complex is the stomata. So water guard cells are specialized epidermal cells that are surrounding or guarding the stomatal pore, and subsidiary cells are guiding the guard cells. They are surrounding the guard cells, and it also helps in classifying. There are various types of stomata we will study in this video, and subsidiary cell, on the basis of subsidiary cell particularly, we are going to classify them. So first, the position of stomata. Amphistomatic, when the stomata occur on both the surfaces of leaves. Generally stomata are distributed on all the surface, leaf, stem, but they are more prominent in leaves. Hypostomatic, when they are restricted to the lower surface of the leaves. Now we have started classifying. There will be, in this video, there will be 5 types of classification by different scientists. So this is A, and we will read up to E. So A, B, C, D, E. In this type you have only 4 types of stomata on the basis of their subsidiary cell. It was proposed in 1950 by Metcalfe and Chalk. These are anomocytic and isocytic, parasitic and diacytic. Anomocytic, the irregular type. Here what happened, you can't distinguish between the subsidiary cells and the other epidermal cells. They are all alike. There are no distinguished subsidiary cells. So it is called as anomocytic, and the families are Ranunculaceae, Papaver, likewise. And isocytic, when the subsidiary cell are 3 in number, out of which one is unequal. That's why the name and isocytic. You can see here, these are the 3 subsidiary cells, 1, 2 and 3, and out of these 3, this one is smaller than the other 2. So it is an unequal cell type, anisocytic. Families which show them are Cruciferae, Umbelliferae, Solanaceae, Convolvulaceae. Next is your parasitic, the parallel one. Here what happens, the subsidiary cells are lying parallel. You can see these are the guard cells in white, and in yellow you have the subsidiary cell. So they are just parallel to the guard cells. That's why the name parasitic. Diacytic, instead of parallel, they are being perpendicular. This is the direction of your guard cell, and this is the direction of your cells. So this is just at right angles to the long axis of the guard cells. So these were 4. Now we come to Stace. Scientist named Stace, and he recognized 2 types of stomata in 1965, and these were actinocytic type, the presence of 4 or more subsidiary cell elongated radially. What is elongated radially? This is your radius, and when cells are like this and they are elongated radially, we can call it as actinocytic. Then cyclocytic. In this type what happens, 4 or more subsidiary cells are arranged in a close long close round around the stoma. You can see here that there are 1, 2, 3. Here you can see there are 3 distinct rounds of subsidiary cells. There are 2 rounds of subsidiary cells. Next was given by Stebbins and Cush in 1961. These are type 1, type 2, type 3 and type 4. This is type 1, 4 subsidiary cells, the guard cells in 4 direction. You can see there are 4 subsidiary 1, 2, 3, 4. 1, 2, 3 and this is 4. This is 1, this is 2, this is 3 and this is 4. Families showing this type of stomata are Araceae, Musaceae and commonly Naceae. Next, type 2. It is surrounded by 2. This is actually surrounded by 4. 2 polar round and 2 longitudinal to the and parallel to the axis. These 2 are long side by side parallel to the guard cells, and these 2 are small polar nodules like round small subsidiary placed lateral to the guard cells, and the family are Pame. It should be E. Next, type 3, the stoma is surrounded by 2 subsidiary cells placed lateral to the guard cell. This is what you find, what you are going to see in monocotyledons. So monocotyledon have this type of lateral placed subsidiary cells. Now the type 4, this is also common in monocots, and here subsidiary cells are absent. Subsidiary cells are altogether absent, and what I have marked is similar. These cells are similar to these cells. So the subsidiary cells are altogether absent here. Now we go to next scientist, Van Cottam, 1970. He proposed 10 types of stomata. So what are these types? Hexacytic. So it is a modified tetracytic type. It has an additional pair of lateral subsidiary cells. There are 6 subsidiary cells consisting of 2 lateral pairs to the long axis of the pore and 2 polar. So you can find 1, 2, 3, 4, 5, 6. 4 are already present, but 2 have joined them. Okay. Again you can see that they have 1, 2, 3, 4, 5, 6. So they have 1, 2, 3, 4 and these 2 extra subsidiary cells have joined them. Next is epicytic. So the presence of subsidiary cell in a diacytic position but lying above the guard cell. So what we have seen in a diacytic, but in this case it is lying above the guard cell. Hemiparasitic, you can see only one single subsidiary cell at one side, this one. So this is called hemiparasitic. This is eupericytic. This is a single subsidiary cell and it is surrounding the guard cell. Copericytic. The single subsidiary cell is surrounding the whole complex, but an additional subsidiary cell is there attached to it, which is partially surrounding the first one. Duplo, we have not missed any. Duplopericytic. 2 subsidiary cells surround. The first encircle the stoma and the second the whole structure. Next is your peritocystic. We have 2 peritocystic and coperitocystic. In peritocystic there is a single subsidiary cell, but it is not surrounding the whole. It is present at the parietal end and one wall is still attached to the, and a small portion is left unoccupied. Here, same thing, parietomycetic, but a extra subsidiary cell is present and it is attached to the first one. Axillocytic and coaxillocytic. Similarly, these are also the same, just here you have only one subsidiary cell, here you have two subsidiary cells, one on the other at the proximal end. Now, this month we will discuss the stomata types and ontogeny given by Dr. Pant, a well renowned scientist, plant botanist. So we will discuss it in next video. These are mesogenous, perigenous and perimesogenous. So we will deal the entire process of ontogeny in the next video. Thank you so much.